So much to the SysMod organizers, especially Matteo and Magna, for organizing this wonderful session today. I was a bit worried that the topic of my talk was sort of out of consistency with the rest of the talks, but as I sat through the presentations today, I saw so many nice tie-ins to what I'm going to be discussing. So I'm really excited to share with this audience today what we've been up to. So a lot of my lab works on cancer metabolism, and through the various centers that were mentioned in the introduction, I've been able to take advantage of the very rich expertise at Georgia Tech and Emory in regenerative medicine and tissue engineering, and it's gotten me interested in this space, specifically how we can start doing a lot more spatial modeling of these processes. So I'm going to just make sure. Let's see, can I? Oh, I'm going to go backwards. I want to get the pointer here. There we go. There we go. Okay. So a lot of my colleagues at Georgia Tech are interested in creating organoids for in vitro assays, personalized drug screening, studying developmental processes. And it's a very exciting time in this field because we've gone from being able to just create one sort of specialized multicellular microphysiological system to a whole repertoire of these various organoids from induced pluripotent stem cells. So as you can see from some of the examples on this slide, everything from sort of intestinal crypts to inner ears, people are studying with these methodologies. However, what is not really conveyed in the publications of this field are the issues of reproducibility and the various cocktails and methods by which these are generated have a very high fail rate. This is a problem if you want to build an industry around these platforms, or like we heard from Imran earlier today, the EPA wants to be able to substitute animal models with these types of platforms. You have to be able to generate that that liver bud over and over again with a high yield rate and not get sort of 1% organoid success rate out of this. So we really have a long ways to go to understanding these multicellular principles that help these populations of cells, which are initially all pluripotent, to differentiate and migrate and form structure and appropriate function. So some of the questions we started to ask for these multicellular systems is how the various environmental cues impact these dynamic changes in phenotypes. Environmental cues can be everything from the media that these organoid systems are growing in, to various cocktails of inhibitors, to neighboring cell morphogen secretion or mechanical tension. How do these local interactions within the networks result in the propagation of spatial patterns? And then ultimately, how form and function emerge from this initial random state? So when I talk to developmental biologists about these questions, they kind of look at me like, and so why are you studying these questions with induced pluripotent stem cells? You should be looking at embryogenesis and how these organs arise in a natural emergent setting. You don't want to kind of create it from scrambled eggs. You want to create it from, the chicken. But I do think there's a real role that these platforms and these in vitro models play because they're so simple. And from a modeling standpoint, you can think of this as just sort of casting this net of sort of initial conditions that then you don't have to have any predisposed memory and positional location information from that initial set in order to start seeing these emergent properties. So luckily for us, there are a number of different ways in which you can perform multicellular computational modeling, and we've heard about some of them today. I'm going to be focused on the agent-based methods, which were very nicely introduced before our lunch break. And the way that we go about doing this, rather than considering the various sort of vortex-based methods or polymer network type methods that are out there, is we think about these individual spheres and how these objects then can interact with one another. We can use them to test the role of stochasticity and reproducibility of organoid culture. We can start uncoupling different differentiation drivers from one another. And those are impossible to experimentally isolate, but our models allow us to uncouple these things and look at the relative importance of them. And as I'm going to be describing today, we can also start to use these models for what I consider designer organoids, being able to create, just like AutoCAD, construct what kind of multicellular system you want and tell the synthetic biologists what circuits are necessary in order to construct those. So what is really fascinating is I'm going to be telling you about various studies. A lot of this is already in the literature, but I'm going to be taking various snippets from our papers to tell you about some of these principles that we've studied. And what's really fascinating is when I talk to my colleagues at Georgia Tech who have studied the evolution of multicellularity from simple organisms, like the lab evolution of yeast into these beautiful snowflake structures, or if you're interested in studying dictyostelium, slime mold, that's another organism that goes from unicellular to multicellular, structures. There are certain principles that keep coming about with these various, in various different sort of kingdoms, right? And some of this is the orientation of cell division and the role that plays, the role of stochasticity, which I've already brought up, the rate of cell cycling, these modes of intercellular communication, and the role of adhesion in the emergence of structure. So I'm going to talk about some of these today. One thing that's very exciting for us is that we're not really as limited as we once were with computational processing. So these agent-based simulations are really coming to the forefront now because you're not as rate limited as you once were before by just sheer sort of CPUs. We still take various sort of considerations in simplifying our models just for the sake of streamlining the computation. So we consider what's the number of cells that's accurate for describing our system, how many diffusible molecules we want, how many parameters do we want to impose, how much morphological complexity is necessary, what kind of timescales and duration of simulations are we interested in? Do we want cell movement, or should we consider a static structure? And that kind of gets back to the timescales of interest. And so our philosophy has been to really describe multicellularity as simple as possible. So you can think of this as a top-down approach. Start with very sort of high-level, simple description and build in more and more processes as necessary. So we focus on the initial loss of pluripotency as a very good stage for doing this, because that's changed from OCT4 positive to OCT4 negative transcriptional status is very, very sharp in its transition. And you can almost think of that like a binary type state change. The other thing that I'll mention is that we've really tried to focus our group on developing quantitative tools for comparing microscopy-based data to models. And to me, this is very central to the agent-based modeling field, because I feel like there aren't enough methodologies for parameter optimization and choice of rules in deciding sort of model selection criteria. So just to kind of give you a preview of some of the things we're going to be discussing today, we've done both 3D modeling and 2D modeling, where we're either interested in spread of a colony on a flat planar surface like you would have in a culture dish, or we're thinking about stem cell aggregates or multicellular aggregates and the various sort of sorting or differentiation that's happening with those. Uh-oh. It's having to think about the slide change here. Let's see. Oh, that's interesting.
To use the arrows.
Yeah, it just jumped for me to slide 2, but it says slide 8 on the bottom. So don't know what to do here. Never had that happen. So that's slide 2.
Yeah. Okay. I think it's one there. Let's see.
We'll just start over.
I think it says okay. Go to slide 3.
No, slide 8.
Okay.
Maybe we go to the next one because that crashed it.
Okay.
Okay, so if I go back there. Okay, hopefully that works online. All right, we're good. Okay, so where I was leaving off, the simplicity of the way we go about this, and then the image-based optimization that we do. So what we tend to do is build our own custom-built platforms. There are a number of software tools out there. If you're interested in sort of a cell POTS type methodology, CompuCell 3D and Morpheus are great supported systems. Physicell is another open software net logo. So there are various ones out there. We prefer to use sort of a physics-based off-lattice methodology with non-deformable agents. And then we subject them to various external forces and consider the cell-cell interactions as spring constants that have sort of defined elasticity associated with those. So we can consider sort of the steric repulsion and adhesion that result from those springs. And then the external forces, such as ultra low adhesion well and the curvature of that well that will force aggregation of that. We can nest simple ODEs in the system. And as we heard from the last talk, Boolean networks are also quite amenable to nesting within individual agents. And as we build up complexity, we are turning to Boolean networks as a very sort of nice way to add in that complexity. So let's talk about some of the simple rules that can dictate multicellular organization. A very simple one is just the rule of when you have 2 cells dividing, where do you stick the daughter cell? And that may seem kind of trivial, but it turns out to have a major impact on the various sort of structural forms that arise from your simulations. And so we found very early on, as we were modeling in 2D spread of an iPSC colony, that one rule alone could give us different behavior. And in my lab, which we also do experiments as well as our computational work, we would observe these various colony phenotypes. In some colonies, you have sort of this like fried egg kind of curvature and doming that it forms as they pile up on top of one another. And then in other phenotypes, you might get these long finger-like projections that really will keep spreading until they hit some kind of micropattern surface change or some physical constraint that keeps them from doing that. And so that alone in our algorithm has a big impact on the behavior. Another rule that dictates the multicellular organization, all of our simulations that I will show you have some element of stochasticity to this. And so if you just consider an initial population of pluripotent cells, you have to have something, start, the events from occurring. So whether that's caused by asymmetric inheritance during division or some kind of stochastic noise, which we know happens with nanog, one of the pluripotent factors, those fluctuations give rise to something sort of pulling the trigger, or tipping the dominoes, if you will. So you have to have some kind of event like that to cause symmetry breaking. And so one thing we found when we were modeling various types of morphogen type-based communication, we could look at various methods in which we were, I've lost my red pointer here, I'll get it back. So we could look at various sort of putative mechanisms that would explain our observed patterning of clusters of OCT4 positive and OCT4 negative cells. And we could essentially compare all of these to the random events. And the random events being kind of the ultimate noises controlling everything in the system. And if you do that, you get these salt and pepper type mosaic type properties. And then you can compare those type of random patterning to the organization that we observe and ask which one of these mechanisms is most appropriate. It turned out for this particular paper that it was sort of this double, one reinforcing and one sort of hindering the state change. So as we look at our comparison between our stem cell aggregates and our model-based aggregates, what we started to notice is that there's sort of this all OCT4 population in cyan that eventually goes to an all OCT4 negative population here. And as you're going through, you start seeing different distributions of these patterns, if you will. You can call them classes if you want, in terms of their radial symmetry or sort of the contiguous type configuration here. And so then the question becomes really a population distribution of these various categories and their temporal trajectories. So the way we handle that is through a segmentation algorithm where we identify positional information within our microscopy images. We use those to create a graph network that allows us to count the number of cells that are within a particular object of clusters, either the positive or negative phenotype. We can look at the size of those clusters, how they are radially positioned within the aggregate, in various sort of membership criteria. The nice thing about that is the dimension reduction then becomes you're going from this network analysis from this high dimensional data set into a lower dimensional one that then is sort of capturing these features of both micro and more macroscopic level organization that can be compared to model simulations. So when we do this, we start, you can plot this in latent space with principal component analysis, and you start connecting the dots, and you can see the type of trajectories you get where you go from that all OCT4 positive population, kind of looping through your latent space into sort of an all OCT4 negative population. And so if you take the sort of the average of the experimental data set, you get some kind of funny trajectory like this. And with our model simulations, we can extract the same network features because we've got these sort of individual cellular agents that are spheres that represent the cells. And so you can go through the same sort of network metric derivation. And you can see that we reproduce that horseshoe shape, but not nearly sort of stretching through our latent space like we would want. Okay, so that's sort of generation one. I'm going to return to this type of approach as I move into our other studies. So another rule that we've looked at is the mitotic dependent intercellular communication. And in order to understand this particular study, what you have to know is that gap junction channels are formed by two hemichannels of connexin proteins. In the case of pluripotent stem cells, our stem cell line expresses connexin 43 during that pluripotent phase. And then the connexin population sort of changes as you move through to more mature phenotypes. So when you have these hemichannels line up, then you have a functional pore. and allows molecules anything less than a kilodalton to easily pass through here. So you have small metabolites that pass through, ions, of course. And so it's really a way of the connexin 43 expression level then is a way of reflecting something about potential permeability and a mode of communication between two adjacent cells. So that becomes very interesting as we start thinking about sort of these graph networks that are being constructed of our positional information. So the student on this project, Chad Glenn, was really interested in looking to see how gap junction-based communication may be used as a driver for differentiation. And he did many microscopy studies where he was looking at how that gap junction property might change as a function of mitosis and day of differentiation. And so he was evaluating this and looking at sort of the cellular distribution, whether these connexin 43 proteins were on the surface where they could form those functional channels, or if they were internalized. And during mitosis, they internalize. It's almost like the cell's closing off. It doesn't want to talk to its neighbors until it's kind of reemerged as, two cells. And then, it expresses them on the surface again and says, okay, I'm ready to open up the lines of communication again. So as we were looking through this, Chad did a number of gap frap experiments where he would load the cells with calcine, photobleach them, and then watch how diffusion from the surrounding cells would lead to a recovery of that fluorescence. And by looking at those recovery constants, he was able to actually reconstruct sort of the cell cycle state across many cells as a sort of a function of that recovery constant. And so in doing so, it was this way of reconstructing sort of this phases of the cell cycle and the relative permeability, if you will, of the calcine. So what can you do with this information? Well, you can think about two neighboring adjacent cells. You can have two pluripotent cells. We know stem cells divide rapidly. So they're rapidly dividing, which means they're opening and shutting those modes of communication more frequently. Or you can have the slower, more mature OCT4 negative cells that, they're cycling less frequently, and they're going to then, have their channels potentially open longer if their neighboring cell is also differentiated. But then you've got these mixtures of a stem cell, a pluripotent stem cell next to a more mature cell, and those phases could be very much out of sequence. And so you can start seeing the complexity that results with things that are synchronized with high cycling, synchronized with low cycling, and then all of the sort of asynchrony that would arise, and this would be reflected in the permeability of material between adjacent cells. So Chad used this to build a model of how differentiation could actually spread and propagate within a colony. And he was doing that by considering a cyclic AMP as our canonical molecule of interest. You have an enzyme that's producing that at a certain rate. If you have that open channel, it can diffuse to its neighbors. If it's closed, it's going to build up and reach a threshold at which it can cause differentiation. And so that's just the simple rule here. And yet it was capable, just by concentration gradients alone, of creating a differentiation sort of propagation wave, which resembled what we were seeing in terms of the propagation of contiguous, non-sort of random loss of pluripotency across the colonies of interest. So before I mentioned to you how we created these different classification, patterns that we could use and look at proportions of those patterns and how we can divide those up with network metrics. What we did in this particular case is we added some transition patterns. Chad created a lot of synthetic images, if you will, or architectures that would reflect these various types. So, this is him essentially taking a paint pen and drawing in colors on a particular, he used lots of different colony shapes, but he can draw something as being inside out or outside in based on the color. And he created these synthetic data sets to train for those latent space network metric type analyses. And just to show you an example of sort of this snaked pattern, when he looks in the microscope field of view, he can start seeing sort of this contiguous sort of loss of pluripotency that's occurring almost like a river running through the colony with that wave of differentiation. So what he was able to do then is train essentially his latent space with lots of examples of the synthetic data set. And he noticed that he could create these various metrics that represent sort of the temporal aspect of differentiation, spatial, amount of randomness. And so his synthetic data set, which is sort of in this sort of transparent coloring, spans a lot of these principal components, whereas his experimental data set kind of lands within a very narrow regime of this. And so what he was able to show with his simulation averages, of course, these had a lot of variance because it's a stochastic type model. But what he could see is that it was really nicely following this experimental trajectory through the latent space. I'm not going to go into the rest of this paper, but we were able to perturb features of the gap junction communication, and the model correctly predict sort of a shift in the location within this latent space. So we're doing better now, generation 2 of model matching sort of experimental complexity spatially. But this was still essentially sort of a hand-fitting operation where we had parameters. We were looking at how that would affect the model performance and iterating on that. I just want to show very briefly an example of how we're starting to use these types of models in synthetic biology examples. So we work very closely with Ron Weiss at MIT, who has developed a cell circuit that uses sort of a stochastic recombinase switch in CHO cells, in which he can add either one chemical inducer or an orthogonal chemical conducer. And based on the concentration levels of either one of those chemicals, he can bias that stochastic switch towards one cadherin isoform or another cadherin isoform. And so you have a way of sort of dialing in sort of probability of a transcriptional output that would affect cell adhesion. And so cadherin 1 and cadherin 6 are the isoforms of interest. The CHO cells don't have either cadherin, so this is a completely artificial system for them. And so you can kind of force it to be sticky with its neighbor in one way or another, depending on sort of the homotypic or heterotypic adhesion. So we're starting from a sort of initial CHO population, and then we're adding the inducer on and forcing it to go towards one phenotype or another. For those of you that aren't familiar with cadherins, these are adhesion molecules on the surface of cells that really kind of rely on kind of Velcro, if you will, with the neighboring cell of the same isoform. So you can have homotypic interactions, which are very, very strong, or heterotypic. So these are going to be weaker if they're between isoforms. So what they were able to do then is dial in different concentration levels of their chemical inducer, and they started observing these globs of either red cells or yellow cells with different types of organization arising from this. And this is at the point that they approached us and said, we really need to understand what's going on here. Can your model help us? And so we started working with them to model the cell adhesion properties. Just very simply, we had to include stochasticity. So this is in the form of thermal noise. We had to then, use known information in the literature about relative strengths of these cadherin to cadherin interactions for our spring constants. And then we kind of allow them to glom together and sort, as that simulation was showing. And in doing so, you can look at this entire matrix of various either sort of red cores or yellow cores and little kind of clusters that occur or no clusters that occur based on the levels of those two chemical inducers. And so across the matrix, our model predictions do quite well at explaining the various phenotypes that arise from these multicellular structures. And what we found from doing this exercise is, first of all, that we thought maybe that there would be a timing component. So if you add one inducer and then wait a while and then do the other versus adding them simultaneously, it'd have an effect. It didn't really. And so our model kind of confirmed that that would occur. We looked at the role of that heterotypic strength on the structures. And one thing we really were very interested in is this role of kinetic trapping. So if you were to jack up sort of the level of adhesion, could we actually get something with a more central core of clustering? And so what we found is actually that kinetic trapping yielded in something where we actually had something that was a disadvantage because it was too strong and things would wall off from allowing sort of escape of particular cells from a cluster. So finally, what I want to leave you with is how we're moving forward with trying to understand this sort of comparison of images to multicellular simulations. So using a particular synthetic circuit that Ron's lab has published, they had observed Turing-like patterns that have formed when they add this chemical inducer of a transcription factor, GATA6, in their pluripotent stem cells. And so we wanted to try to reproduce those patterns. And in doing so, we created agent-based models with very simple switches from sort of a nanog high, GATA6 low population to GATA6 high, nanog low population. And the big question here is, all right, so we can build a model like this. How do we know if it's good enough? And the big question is this sufficient? How do we evaluate model performance? And how do we design sort of new outcomes from this? So for the past few years, we've been working as part of a NSF Simons Foundation Center that brings together mathematicians with biomedical researchers. And so working with mathematicians that are experts in topological data analysis, TDA is something you can use for very high dimensional data sets. And what you're looking for is structure in your data in the clouds. And the way you're doing this is by creating kind of larger and larger spheres of potential interaction between the data points in the data cloud. And the point at which these spheres begin to intersect, that's the birth of a cycle. And you keep building that radius up until you have full closure of the circle, and then that becomes the death of the cycle. So the birth-death processes are reflecting cycles in your data set. And so, you're looking sort of at these distances of a whole. And then from that birth-death process, you can flip this on its side and you get what looks like a little mountain valley, mountain range, and that's your persistence landscape. What's really nice about that is that it allows for statistical analysis to quantify differences between patterns. It provides very interpretable metrics, and you don't have to have a large number of training sets or deep learning approaches in order to extract nice features in your patterns. So this has been used before to look at developmental processes in an agent-based model of zebra stripe formation. There's a wonderful paper in PNAS on this. And we wanted to apply it to our microscopy data, and we found that doing so is actually very technically challenging. And so, we spent a long time working to develop a pipeline for processing experimental images. And this is something that's been used to sort of analyze stock markets and such like that. But what we're trying to do here is just take simple XY coordinate information and looking at these sort of various categories of high versus low GATA6 populations. So we start with segmentation. We've got different classes of high or low nanog, high or low GATA6. And from that, we can create with Delaunay triangulation sort of a graph of our positional information that allows us then to perform those birth-death processes. And so we look for the cycles within those, and we get these persistence landscapes from this. And then you do this many times and you get sort of averaged information about this. So we use this to analyze the different types of antibodies, which would allow us to evaluate either endogenous GATA6 levels or synthetic GATA6 levels. And in doing so, it was really sort of, does this image right here look any different than this image? Well, there's a little more red, perhaps, but topologically, is that different? So we can use these types of pipelines to statistically analyze and discriminate between these. And what we started to find is that sort of that synthetic circuit actually creates bunching of the cells with larger persistent holes. And that's actually something that's very interpretable. It's not just a sort of a feature that you plug into ImageNet and you say, yes, these are different. It's telling us what's different about them. And what was really sort of exciting is it gave us sort of a putative mechanism to start going after experimentally in terms of how the motility of GATA6 high cells may be different and the chemotaxis that could be taking place here versus the endogenous low levels of the cells. Okay, so I'm running out of time. I'll just say we're starting to use these to now make direct comparisons between our agent-based models and our microscopy. Now we have a way of optimizing based on these persistence landscapes that should allow us to, no matter how different the various individual configurations are, they're saying something here that we can actually look at the differences and try to minimize those differences with our parameter optimization. So I just want to leave you with the fact that there are so many options in the culture and construction of organoids that computational models really are at a place where they can start streamlining reproducible organoid manufacturing. And even the design of new synthetic biology multicellular circuits, which I think mammalian synthetic biology is now at that place, can take place here. Finally, these robust tools for comparing multicellular images and agent-based modeling outputs in an interpretable fashion is going to be really useful as we enter the age of spatial omics, like you've started to hear about at this conference. And it helps us move from just simple classification tools into revealing sort of pathological mechanism. We can really start getting into how those various high-dimensional data sets in spatial organization are playing a role in disease. So I would just like to thank my current and former lab members, specifically Chad Glenn, Douglas White, and Unbi Park for all their work on this, and our funding sources. And finally, I just want to leave the audience here and online with sort of a preview that in winter 2025, myself and Eberhard Voigt will have a new textbook out. It's great if any of you teach systems biology in your classroom. We've really worked hard to update this because this is a rapidly changing field. Staying true to the SysMod, community, this is not a machine learning textbook. It's really focused on design principles, nonlinear dynamics, and understanding features of complex systems with differential equations and the tools that are out there today. So with that, thank you for your attention, and I'm happy to take a few questions.
Thank you, Melissa. That was a beautiful talk. I would like to open the room up for any questions.
Hey, thank you for a great talk. And I have a couple of basic questions.
Yeah.
And the first one is like, to my understanding, like agent-based model is more like a discrete model. They might lead to like chaos, or in the end, if we try to solve the steady state. But what brings you to use like agent-based model over like something like stochastic Langevin equation or like recipe function to model the like the all the change of the sphere or the cells. And second question is like, to my understanding, I wonder the scale of the diffusion rate for the like the first part of the model, because the most of the protein, they can diffuse really quick, like in nanosecond, it can be just homogeneous, and instead of like building like symmetric like breaking like patterns. So how do you like define the rate for diffusion, and how do you use like the fitting method to find the proper like diffusion rate for that compared to like protein production or something like that?
Great questions. So let me start with the first one. So in terms of the approach to just using PDEs, for example, to describe morphogen fields. One thing I think you can certainly do that and just look at sort of field effects and assume that the cells that are in those locations of the field are then going to have the, whether it's sort of a bandpass filter or a threshold that leads to a state change, and say, all right, there, I'm getting my French flag because I have, these stripes that occur. What that doesn't take into account, and we can create field effects by having the secretion of those molecules. So we are using the Langevin equations, in a numerical sense when we're accounting for secretable diffusible factors. But what that doesn't allow, if you just do the field effects alone, is any kind of chemotactic movement that the cells are doing. So if you have cells that are then migrating towards a gradient, that, if they're positionally static, then they can be instructed, okay, this is, what we're supposed to do. But if there's any type of migration that's occurring with intercalation or a spreading along a surface, or some of the cell sorting that takes place, you're going to miss that because you're considering, just those sort of continuous systems. So the second question you had was about the various diffusible ligands. I don't recall the exact, diffusion coefficients that we used in that study. However, as I recall, there were several sources in the literature of LIF, which we thought was a putative ligand for the mouse embryonic stem cells that we were working with there. And I think FGF was the other one that we used as potential diffusion coefficients. And then the sort of the long range versus short range distances, I think if we were to put this on an order of sort of first degree versus fourth degree cell separation, that would give you an idea of kind of on the scale of under 10 microns versus a 40 micron distance, if that's helpful.
Yeah, thank you. But I actually have another question. It's like, have you tried to solve like the steady state for like those model? I know like it's kind of hard for like agent-based model to find the steady state, but if you don't know like steady state, how do you know like if it's just a spatial case for the agent-based model, the solution, or it's just like the stable like solution for all kind of like different like parameter or different like situation?
That's a great question. So with the self-sorting model, we ran it out a really long time to ask if we were just looking at a narrow window that reflected the experimental setting, are we missing something? And you take it out long enough and they'll all kind of group into a particular steady state distribution. We didn't see that with that particular model. What a mathematician we've been working with, Daniel Cruz, who's now at University of Florida, he's really interested in looking at steady state distributions of agent-based models. And so, can you actually calculate that without doing the numerical simulation? And I'm not a mathematician, so I don't understand all the theory behind it, but the answer is yes. I think, you can estimate statistical distributions. So that's something that he's been working on, and I think is kind of at the forefront of the modeling theory, if you will.
Also, it's like they are not trying to use like bifurcation or like phase trace to find a state. Say they try to use like statistical like method to estimate the result.
Mhm.
Oh yeah,
Yeah.
that makes sense. Thank
Yeah.
you so much.
Yeah.
Thank you, Debbie.
Thank you very much for this great talk. I had a question about your, when you show the difference between the healthy cell and the cancer cell through your simulations. Are you then because then you make a perspective about synthetic biology, are you then able to suggest which could be then the mechanism or which are the parameters that could be then influencing, reversing the cancer state to the more healthy state?
Okay, I'm not sure which you're referring. Are you talking about the CHO cell study?
Yeah.
So that is sort of an irreversible state change that takes place. So when you add that recombinase switch, it's going to express the cadherins, and it can never return back.
Okay.
So it's a unidirectional state change.
Can you plug in somehow in a sort of synthetic biology view? Can you plug in, for example, some feed forward or some positive or negative feedback loop within your system that actually can help you to revert the state or switch it to another state?
Absolutely. Yes, we haven't done that, but I know that that's within the repertoire of tools that's possible, and certainly one we're interested in. Yeah,
Thank
that's
you.
a very
Thank you
good
very much.
question.